So this third video in G02 looks at genome assembly approaches, and we're going to talk more about the ways we go about this now. So when we think about this problem of repetitiveness, we can do things like shotgun sequencing, where we take a whole bunch of fragments and sequence them all together, is that we have these repeats. And there are different ways to just try to get around this problem to how, in terms of relating the reads to each other. Paired end reads are one way of doing that. For certain specimens, that may not be required. So basically, we have a forward read one that goes in this direction, and then a reverse read that goes in the reverse direction. That's why they're commonly known as forward and reverse reads. And then there's an insert between them, where this whole segment of DNA comes from a single fragment. And that insert may not be a known distance, but it certainly is a distance that can be estimated. So that means we have a bit of known DNA here, and we know that they're a certain distance apart, an expected distance apart. And that can mean that we can identify situations where either one reads maps only, or situations where they're very, very far apart or misoriented in some shape or form. So if we have our reads aligning like this, then we know something has changed in our sample compared to the reference. Our reads are typically structured like this. So we have our barcode located down on one end. So we want to get rid of that. Same with the adapter. And then we'll have our reads there with the insert size somewhere in the middle. So if we have single end reads, our read might span this length. The lengths typically range from, I think, from 100 base pairs all the way up to 300 base pairs. Though, of course, not all of those bases may be used in the process of assembly because we may have done QC to get rid of the low quality bases. For paired end reads, the other reverse read comes from the same fragment of DNA. So some sort of insert in the middle. For many libraries, that may be of the order of 300 bases, but that does vary. The longer this insert size, the fewer sets of pairs that we will get at the end. So it's the sort of a balance, because we also want a longer insert size to get a wider resolution for the structure of a genome coming from a longer total DNA fragment size. So there are many different read types, and this image is taken from this paper down here. So we have our forward primers and our reverse primers known as F and R. And we might have our long double-stranded DNA fragment up here. We might toggle go through cycles to prepare the library and generate these reads. So you can see the reverse and forward primers here. And this can generate what's called a mate pair library. And that, in essence, means that the insert size is now going to be very, very long, something like maybe 1,000 bases, or maybe indeed 2,000 bases. However, if we've a really, really long insert size, the number of reads that we will obtain at the end of that process is going to be far, far fewer. And that's simply due to the library preparation process. Most of the fragments will be much smaller than that. So that kind of is a third option aside from single end reads, where we have our read and no insert. And the same with the reverse reads, where we have the primer attaching to the read, and our reverse primer there. So this kind of brings us back to our main situation we had from the FastQC practical, where we had Illumina reads that are short, numerous, and accurate. Typically, they have more PCR that can be a source of bias, and they resolve each base very, very well, whereas nanopore reads are generally fewer, they're less accurate, but they're long and they give good scaffolding information. So to kind of summarize these kind of differences and how they can be differentially used in assembly. And ideally, we want to combine them together. So you consider this, that if you have two contigs here, like contig A and contig B, what we would like are long reads, and this could be nanopore reads or mate pair reads, or any kind of read that will bridge from this region all the way over to this region here. That means we know if we don't know the intervening sequences between A and B, we at least know the length of the gap. That's very useful. And we can use this kind of long read information that if we can bridge them together with a known gap, we can remap reads to these gap edges at times and actually bridge some of that gap iteratively after we form the genome assembly. So there's a lot of work that can be done after genome assembly to try to improve our assembly further. So if we look at this diagram, what we can see here is that it's showing different short reads that can be merged into contigs based on the sequence overlaps and the quality scores associated with them. We can then add on long-range links on there and go from contigs to scaffolds to chromosomes in that way. So for our host genomes, you might find that there we get many contigs, we got many scaffolds, and hopefully we'd like to resolve them into simple chromosomes. But that obviously is challenging. In our practical, we're going to be dealing mainly with various libraries, and some of these we saw before. They might have something like 300,000 reads with a read length of 101 base pairs and insert size of 170. That's a good example. And that gives us a graphical overview of the length of these reads at the forward and reverse ends, and then the insert size in between. Of course, this value is actually a mean value. So if we look at these particular libraries and look at the spread of insert sizes, for any library, it'll look similar. You can get a range of values. So this is for the library no name. You can see there peaks at about 190 is the mean right there. But you see there's a spread of insert sizes. And in some ways, having longer insert sizes is useful there. And the same for G3682. It has a longer average insert size. So you can see that assembly is probably going to work better. And then we see G3771, and we know from the last lab that quality was not great. So I wouldn't be sure how this is actually going to assemble. What we can also use as well in our assembly is that we can remap the reads back to it. So if we imagine that we have a reference sequence shown here in blue, and this might be an assembled contig, we can map the reads or segments of these reads to these regions and see how similar they are to each other. So for example, here we've got T bases all the way through, suggesting that that's the consensus base there. And note that all the reads are similar to the blue one at the top. So that suggests that the blue one reflects the consensus of all these different reads. But we might want to identify situations where we have mismatches where we might have to think about what type of assembly are we making. So by counting up all these bases here, T for this T base, what we can see there is that we have 1, 2, 3, 4, 5, 6, 7 reads all together there. So that gives us a measure of the depth, the read depth at that particular site. And a read depth operates on a per site basis that you can see for this, let's say this G here, we have 1, 2, 3, 4 reads only. So this one is 7, and this one has a read depth of 4. So read depth will vary across the genome depending on the number of reads that were sequenced. The more reads that we have, the better resolution we will have of the true consensus sequence. And a simple way of calculating that is to take your expected genome length and the number of reads and determine the coverage as the length of these reads multiplied by the number of them divided by the genome length. So that kind of gives you the expected coverage, bearing in mind that this will vary a lot across the genome. And you'll note that conventionally, when people talk about fold coverage or read depth, they put an X at the end to indicate that it's 2X coverage, 5X coverage, or 100X coverage. So we can think about how these reads can potentially resolve and where they might actually work well in resolving a genome sequence or where they may struggle. So one example here from this paper down here is where we have a bunch of short reads in yellow and long reads in brown, and we're trying to resolve this gray and red region where we have repeats here. Here you'll note that we have 2 repeats. So the short reads collectively may struggle to identify which repeat is which. If we have this read inside here with the green X, you see that its start and end are inside that repeat. So we wouldn't know where to place that here or in that position there. Whereas this one here with the circle has a part gray and part red. So we know that that's the start, and vice versa. For this one here, we know that this is part in the red region and part in the gray region. So you can correctly place that. If the repeats are, if we don't have any reads that span this region or here, long reads can tell us about the length of that region. So we might resemble these contexts here with the short reads only, and then have some uncertain gap here that resolved by the long read to create one assembly with our repeat inside. If we look at another example, we may have a repeat where the long read is over that repeat, so it doesn't complete over to the other side, as you would hope, which means that the repeat is not a clear length. We can't really at least resolve that as a repeat, because we'll know that this read starts in the long read and goes into the repeat. And this one does it from the other side. It goes from the long read into the gray area, and vice versa. This one goes from the repeat into the gray area. So note this is a region of determined reasonably accurate size length, but they'll all be on different contexts. So we have 3 contexts in this situation. So that's why we like sufficient coverage for our long reads to resolve all these repeats. But if you have many repeats, that can be difficult. In the same way as long reads, paired end reads can also help resolve regions that are of interest. So if your short reads alone will resolve these 2 contexts here, the paired end reads can resolve this bridge here and help us resolve at least the size of this gap. So they don't tell us the sequence's size. So this will just be denoted as a bunch of ends, but ends of a known size. And that is useful in determining this region. So if we're trying to finish this genome, we could design primers that can sequence out from either edge. So resolving repetitive elements, it could be a transposable element or whatever you are trying to resolve. We have to bear in mind that we may have a reference genome in green and repeat here with a particular length, and our context on either side, and our assembly might be in blue. That if we got repetitive elements that are present many times in the genomes, that can be difficult to resolve exactly where it is. So we may not be able to resolve this fully, depending on whether reads are spanning this region or not. And if they are fully inside this region as well, that may mean it can be difficult to quantify the length unless we have paired end reads that bridge over, or we have long reads that bridge over as well. So what we can see as well, that is that if our repeat is at an end of a reference and far from resolved regions, so this might be some sort of unknown gap, that can also mean that can be misplaced in assembly. So we may have a low complexity region here that can be hard to resolve, and that can mean also that the reads that are taken from here end up aligning this closer to this blue contact one than actually is the reality. Finally, in this area, we have more advances that we're going to touch on later. One of them is graphical genomes, and the ideas behind there. So instead of having a reference genome of a sequence, we have a reference graph containing the mutations that we observed so far in our population. So the graph might be derived from pre-existing studies or other work where that's available. And what we're doing here is we're mapping our reads onto this graph to determine what regions are similar and what regions aren't. So you can see that we might have 2 mutations, A and B, that are have different paths in this graph representing sequences. So this pink path is a sequence, and this green graph is another similar sequence. Vice versa for red and yellow. And we want to see, do our reads map to these or not? So this is similar to a linear genome alignment, but at least tells us which paths in the graph our reads should map to. And that can mean then we can identify mutations based on sequence matches that differ from the reference sequence. In the same way, if we have a plot of reads here shown by different colors, and each of the 7 colors represents a different sample, we can construct this variation graph containing the mutations, like maybe a snip here between G and A, and then a conserved region before deletion present in certain samples, a conserved region, and then an insertion in certain samples, and so on. We can document this graph a bit like a tube map, going from place to place to place. And then think about these as genome graphs, where a set of genome graphs represents each of the different samples that we have. So this red sequence goes from G to the conserved sequence here. It lacks that deletion. It has this conserved sequence. It has the insertion and has the final conserved sequence. And that contrasts with some of the others down here, where for the yellow one, we have the A allele and then the conserved sequence, and then it has the deletion, and then it has this conserved sequence, it has the insertion and the conserved sequence at the end. So this genome graphs can be extracted from variation graphs where genome graphs represent different samples. So that concludes this video on assembly approaches. Please see the next video on de Bruijn graph assembly.